Hello, fourth grade scientists. This is Scientist Cook, and I am going to walk you through what we're going to be doing for your bean dissection. So before we start the whole dissection, we need to do a couple of things. Number one, you know that you took your materials out of the bag, and it's going to, this sheet will sort of tell you what we're going to do. We're going to dissect or take apart a lima bean. The purpose of that is to find out about the parts that it already has that help the lima bean grow into a lima bean plant. That is going to help us answer the essential question of what are the parts of a plant that help it survive, and what are the functions or what are the functions of each part of that plant. So before we begin, you have this index card. You're going to want to gather a little cup full of water, about an inch of water, and you've got your lima beans. And before we even begin, I want us to kind of look at the lima beans and think about what it is that we can observe, because we know that a scientist is an observer. All right, we can use our senses, our eyes, our ears. I don't know if a lima bean, if we can hear anything, feel the texture, use our hands, maybe smell it, see what's going on there. And so I want to make at least 5 or 6 observations about this lima bean before I soak it for our dissection. So I'm noticing that it's kind of white. Okay, I guess I could measure it. I'm going to use a benchmark measure. And I know that my thumb is about an inch from the tip to there. So it's about an inch long, about 1 inch long. It appears to be, it's like curved in a semicircle. The outside is smooth. And I'm jotting all of this down on this index card that was in the kit. There are some more observations, and I would love to see what other observations that you make on your card. After I've made my observations of the beans before they soak, I'm going to take these beans and I'm going to place them into the water, because they're going to need to soak in order for us to do our dissection successfully. They're going to need to soak for about 2 hours. Even longer is better, but a minimum of 2 hours so that we can really look inside and find all of the parts. While they're soaking, probably not for the whole time, because that would be a very long time. While they're soaking, your job is to now study about the different parts of a bean seed, because when we go inside the bean and look at it, we want to see if we can find all of these parts. So we've given you these sheets. So this is pronounced epicotyl, and the epicotyl develops into the plant's first leaves. So we're going to see if we can find the epicotyl inside. The hypocotyl develops into the stem of the plant. So when we go inside, we'll look and see if we can find the hypocotyl. The radical develops into the root, radical root. This whole thing, all these parts are the embryo, which is a young plant developing inside a seed that is not yet germinated, which means sprouted. Then there's the cotyledon. The cotyledon is the stored food for the young plant. And then there's the seed coat around the whole seed, which protects the seed. It's the outer coating. So here's a labeled version. On the next page is an unlabeled version, but we're not going to look at that until after the dissection. But here we have seed vocabulary cards. So what I did is I colored the different vocabulary cards, and I'm going to cut them out. And if you cut them out, you cut along the dotted line, okay? And then you just fold like this. You do not cut, you just fold across the side, okay? Or you could cut all of them, and you could make yourself a game, which is like a matching game, okay? So again, I enjoyed coloring all of the different parts of the seed, and I'm going to study those parts so that when I do actually dissect the seed in our next video, I'll see if I can find the parts.